I don't think they are able to watch the stream like that. So I'll use that one.
Have to see the screen. Sure. Let me see if Andy's there. You can't see the screen.
Now they can. So then do have the basketball. It's like you like to watch my puzzle. Okay. And I don't have to watch it before it comes live. You just have two buttons.
No, eventually you're still gonna have to wash it.
Yes, my brother is confirming that they can see it right now. They can see it. Great. Sorry? I think so. Let me ask them.
Can hear you.
Okay, so I guess we're at 12:05, so we'll start. It's my pleasure to introduce Alberto Perez from the Technical University of Madrid, which is in Spain. He's in the Department of Audio-Visual Engineering and Communications. He is a CA and he's entering the last year of his PhD. So that's 4 years.
4 years in Spain, yes.
Alberto did a BSc in Sound and Image Engineering and Telecommunications, followed on with a master's, and really did work on the Internet of Things, and then moved to the dark side of biomedical imaging, where his PhD research is on in vivo human brain tissue imaging and classification, with the goal of being able to identify tumors and tumor margins, I believe. He's working with Professors Eduardo and Cesar. Alberto received a 5-month fellowship to study in our group. He has some interest in spatial frequency domain imaging, and he hopes to add that to his multispectral or hyperspectral imaging. So he has been here since the start of May and will be here through October. So if anyone wants to interact with him outside of this meeting, he'll be around BLI for a few months. So with no further ado, then please welcome Alberto, and thank you.
Well, before beginning, thanks, Dr. Kennedy, for such great introduction. And also, I would like to thank Dr. Durkin and Dr. Choi for inviting me to come here to BLI to do my PhD stay. So today I'm going to talk about the research that I conduct, or we conduct in the research center I'm from in Spain. We have many different research areas, but the most relevant one for BLI is probably the one that we conduct with in vivo human brains. And today in this seminar, I'm going to talk about how we are able to identify brain tumors, more specifically the margins of the brain tumors of in vivo human brains, and how we are able to use a multimodal approach in which we are able to integrate the information from machine learning models when classifying hyperspectral data, and how we fuse that information with using depth information, and potentially using that all of this information to merge everything into magnetic resonance imaging. So what I'm going to talk about today is first about my research center and the technologies that we use, really briefly. Then I will talk about the prototype that we have developed to gather a really huge database that we have that is growing in a weekly basis, and also kind of how we are building these systems. Then I will talk about how we are able to fuse hyperspectral imaging and the machine learning classifications on how we want to get that information into the magnetic resonance images and how we want to fuse that information using depth information. Then I will talk about the usage of ensemble learning, which is a branch of machine learning for biomedical applications. Here I will talk about how we are able to use ensemble learning for brain tissue identification and classification, and how we've been able to use these techniques that we've developed for SFDI data for burn wound severities. And last but not least, I will talk about the prototype that I've developed and the main reason why I'm here today and doing my PhD here, which is a SFDI and hyperspectral image prototype that I'm currently using in the OR for imaging burns in pigs. So yes, I will introduce everything in the future slides. So yes, let's start. So I'm from Spain, and basically my research center is in Madrid. So what you can see here is the campus where we work in and where we conduct all our research. This campus is from the Technical University of Madrid, which is the one that I'm working with. Inside this campus, there's the telecommunication school in which we teach classes and we conduct all our research. And also inside here, inside the red rectangle, we have the research center, which is where we conduct all our experiments and all our research, basically. And the name is Research Center on Software Technologies and Multimedia Systems for Sustainability. So in our research center, we work with different technologies, and the main ones that we use are focused on brain tumor delineation. And this is a really important thing because, especially in really infiltrative tumors, it's really hard to differentiate where the tumor is regarding the healthy tissue. So the tumor itself gets into the healthy tissue, and it's quite difficult even for neurosurgeons to delineate where the tumor is. So that's why we want to use different technologies. The first one and the main one is hyperspectral image and video. For that purpose, we have two hyperspectral cameras. One is a snapshot camera and one is a line scan camera. I will talk about in more details in the next slides. Then we use immersive video. So we use depth cameras to obtain depth information of the scenes. And we are able to use this technology to fuse the hyperspectral data into other kinds of data, such as RGB or potentially magnetic resonance imaging. And of course, we are starting to work with MRI and PET systems. We are quite new with these systems yet, but we have received some systems in our research group, which are an MRI and a PET systems, and just we are starting to learn how to work with this and potentially use it with rats, mice, and small-scale animals. And just if anyone is interested, our MRI system has 9.4 Teslas, so it's quite powerful. And once we are able to learn how to use these systems, we will try to merge all that we know with MRI. So now I'm going to talk about the prototype that we have developed to acquire data inside the operating room. So we call that the Slim Brain prototype. And this prototype was built thanks also with the collaboration with a really important hospital in Madrid, which is Hospital Universitario 12 de Octubre. This hospital, we've been collaborating with this hospital for 5 years now, and we expect to collaborate for many more years in the future. And this is one of the latest prototypes that we have developed. As you can see here, this is number 3. We have developed so many prototypes throughout these 5 years. And just so that you can see here, we have the processing station, which is basically a really powerful computer with really powerful GPUs and CPUs, so that we are able to process all the hyperspectral data, classify with our machine learning algorithms, control of all of the systems, and so on. Then we have a light source, which we use. It's a broadband light source, which we use to illuminate the brain surfaces, and we mainly illuminate in the visible and near-infrared spectrum. And here what you can see is the main components of the Slim Brain prototype of the third one. So as you can see here, we have 3 sensors, 3 imaging sensors. The first one is hyperspectral snapshot camera. This snapshot camera is quite powerful because we are able to acquire the spatial and the spectral information in one single exposure time. So that's really helpful and really powerful because we are able to acquire real-time videos, hyperspectral videos, and that's really useful for diagnosis or inside the operating room. However, we also have two other systems, two other imaging systems. The other one is another hyperspectral camera, but this time this one is a line scan camera, which provides a way more spatial and spectral information compared to the snapshot one. However, it requires a motorized linear states to perform the scanning of the brain. And we use this camera to double check that the information that we are capturing with the snapshot is good enough for our purposes. And also we have here a LiDAR in this case, in this prototype, which allows us to get the RGB information of the brain as well as the depth information. So basically how far away or how close away are we from the system to the brain surface or anything around it. So yeah, this might be a little bit gross, but sorry about that. I know you are having lunch right now, but whatever. This is basically the system being used in an operating in a surgical intervention. So yes, we go on a weekly basis to gather information from in vivo human brains patients that may suffer from different pathologies. Maybe they don't suffer from pathologies, but they have aneurysms or other things that are suffering from. So yes, this is basically the prototype. This is the latest one. We have reduced the size of it. In the past prototype, we had 2 screens. Now we have 1, and we are constantly trying to reduce the size of it because the smaller it gets, the better for the neurosurgeons because they are they always ask, can you make this thing a little bit more small? So we try our best to do that. It's quite complex because we have so many systems inside, but we try our best, and this one is working perfectly, and we are using this one lately for these past 2 years, and it's working really, really well. So with these prototypes throughout these 5 years, I've already mentioned, but we've gathered a really huge database that we've called Slim Brain database. We have described all the data inside this database in a paper. This paper is yet not published. However, there's a preprint version available. So if you want to take a look at it, you can just scan with your phone this QR, and we'll redirect you to the preprint article. So yes, if you also want to have more information, this is one of the websites that I've developed with my colleagues. It's a website where you can find more information about what we are researching and what you can find in the database. And just as a summary, we have hyperspectral, RGB, depth images and videos for more than 200 patients. This again is growing on a weekly basis, and we have patients suffering from different pathologies, not only glioblastomas, which is the main tumor that we are focused on. And yes, thanks to neurosurgeons in the hospital we are collaborating with, we have over 1 million labeled pixels from healthy tumor, blood, and even meninges tissues. And we use these labeled data to train our machine learning algorithms to perform these classification maps, as you can see here. So again, we have lots of data. I know I've been saying that for so long, but there's an issue because if the database is really huge, it's really difficult to find the right information that you want to conduct your research. So for that reason, I also developed this website interface in which you can have in a single table all of the information that we have gathered. So we have which patient have been captured with which camera, with which illumination, with which prototype, which pathology the patient is suffering from. And it's really an interactive thing to use to search, filter, and download patients' data, either hyperspectral, RGB, or depth information, images or videos. Yet this is not available, but once it's available and anyone that can have access to this will be able to use this table. So now I'm going to talk briefly one of the things that we are trying to do, which is basically compare both hyperspectral cameras that we have. We have the line scan camera, which gathers almost 400 high spectral bands from the visible to the near infrared spectrum. And then we have the snapshot camera that is not that powerful in the sense that it doesn't capture enough or many spectral and spatial information compared to the lens scan camera. However, as I already mentioned, we are able to obtain hyperspectral videos from this snapshot camera. So one of the experiments that we did, and actually these images are available in the database, is capture in vivo human brains, and we ask neurosurgeons to place sterilized rubber rings in the brain surfaces to identify different tissues. So for instance, what you can see here inside the green rings, neurosurgeons are saying that there should be healthy tissue inside. However, if they are placing a black ring, they are mentioning that there is tumor or a pathological tissue inside. And this is really helpful because this allows us to double check if our machine learning models are actually predicting what they are expecting from by placing those rings. So to compare these cameras, I tried to compare them spectrally so that we can make sure how similar the measurements are from one camera to each other. And what we've done is basically get some pixels from the insides of the rubber rings, compute the mean spectral signature, so the diffuse reflectance across the wavelengths. And we've performed some comparisons with some spectral similarity metrics using RMSE, spectral angle mapping, and other metrics. I won't get more into the details, but again, this is one of the things that we are researching, and with this information, we wanted to look for biomarkers within the hyperspectral measurements. By that, obviously, with the hyperspectral cameras, we are not able to acquire or to obtain the absorption coefficient right now with what we have. However, what we try to do is get the reflectance, apply this equation, this expression, and obtain the absorbance, which allows us to see the peaks of potential some chromophores, absorption chromophores. So for instance, these are some of the results that we obtained. These are the tumor spectral signatures gathered from both hyperspectral cameras. And you can see in different rectangles, the different absorption peaks from different chromophores. And although we are not able to see the concentration of these chromophores in our measurements, we can see that we are finding some peaks that might indicate the presence of these chromophores. As you can see here, for instance, we're able to see some hemoglobin peaks in the measurements. And also here we can see some peaks from some of the cytochromes. So again, this work is not published yet. We've sent this for review, and if you want to take a further look, you can read this preprint version, which is available here. So yes, let's continue. So now we are approaching, I would say, one of the most important things that we've been able to achieve in our research group, which is basically how we are able to fuse the information from all of the different sensors that we have in our prototype. So we get the hyperspectral images, we train machine learning models, and we get more hyperspectral images, and then we classify those hyperspectral images. But we want to merge that information into the magnetic resonance images. And I'm going to talk about why that's important. So the main thing is that when neurosurgeons remove the bone flap to expose the brain surface, the brain, because of how it's conformed, it shifts towards the opening. So that's an issue because they use magnetic resonance imaging before going to the operating room to locate where the tumor is. But because of this brain shift, we no longer know where is the precise location of the tumor. So one way to solve that is by using hyperspectral imaging and fuse that information that we get into the magnetic resonance. So the magnetic resonance images are what the neurosurgeons are, what they really know how to work with. They don't know how to interpret machine learning classifications or whatever. They don't know how to. they only know how to use magnetic MRI images. So I'll talk about that in future slides. So again, we have the hyperspectral snapshot camera. We are able to gather hyperspectral videos, and we are able to train machine learning models and generate this kind of classification maps. So in different colors, we are identifying different brain tissues. In green, we are saying the tissue is healthy tissue. In blue, we are saying that there are some blood vessels. And in pink, it would illustrate that there are some meninges. Even though this example does not illustrate tumor, we should be able to see red pixels in this classification map illustrating where the tumor is, where the pathological tissue is inside the image. So I think, yes, you can see, right? Like you can see the brain is moving, right, a little bit. So this means these are videos. I promise these are videos. These are not images, okay? So again, we have hyperspectral videos and hyperspectral classification maps. Then we use RGB-D cameras, which stands for RGB and depth cameras. And we are also able to gather videos from the surfaces. So this is an RGB video of one of the patients that we have in our database. And what you can see down there is a depth map, which basically indicates in a grayscale how far each of these pixels are corresponding to the system, to the prototype. So now, one of the things that my colleagues have been able to do is to fuse this classification map into this RGB video and create a 3D immersive tool potentially that could be used for neurosurgeons. So you are going to see the result right now. So what you are able to see right now is a point cloud in which we are able to fuse the RGB information, and on top of that, we are able to see that frame, which is from the hyperspectral camera, and how we are able to classify that in real time. So again, this is a real-time video that we can show to neurosurgeons. And this is really helpful and allows us to navigate through the scene, zoom in, zoom out, and basically merge this kind of information, which is really, really powerful. Again, if you really want to know more about this, there's a published paper available here if you want. But yes, let's keep moving, because this is not where we want to stop. We want to fuse, the end goal would be to fuse that information, that machine learning classification map, into the magnetic resonance, because neurosurgeons are more familiar using MRI instead of machine learning classifications. So again, we have neurosurgeons have these magnetic resonance images of the brain before going into the surgery. Then we are able, in the surgery, to gather this information with our camera, create these classification maps to illustrate where the tumor is. And we would like to fuse and register in real time the machine learning classification from here into the magnetic resonance. So we are here able to detect where the tumor is. Trust me, tumor should be there, okay? And we want to place that into the magnetic resonance. That way, we try to solve the brain shift issue that I was mentioning before, which was basically the brain moving towards the opening of the skull. So now I'm going to show you how we want to do that. And I'm going to show you a really cool video, a really cool simulation of what we would like to do in the operating room. So this is a 3D video one of my colleagues in Spain did, basically illustrating how we want to conduct this registration. So during the operating room, during the surgery, before starting, they place the patient in the bed, and they have to register the position of the head of the patient with regard to the magnetic resonance, so that they are able to have a 3D model of the magnetic resonance, as you can see here. This is kind of a simulation. Once the magnetic resonance is registered with the position of the patient, we do then perform a tracking part in which we track with our depth camera the position of the brain or the patient. And then what we try to do is, now we have the magnetic resonance volume, then we have the classification, the hyperspectral classification in the brain surface. And then we want to perform this registration. And you may ask, how are we going to do that? That's really difficult, and it is really difficult, but we've been able to get some results, and the results are quite impressive. In the 1 month ago, we published an article describing how we are able to fuse this in a preclinical situation, not yet in the operating room. And this is the paper. I won't get much into the details because it's quite hard, and it's a really tough thing to understand from my point of view. But it's really powerful because the errors that we are able to obtain are less than 1 millimeter. So that's really, really, really precise. So what we do is, one of my colleagues have developed a really small prototype, which is this kind of thing right here, where we have a depth camera, and on top of it, we have the hyperspectral snapshot camera. So with the depth camera, we obtain the geometrical phase of the patient, or the volume in this case. And with this depth map, we use ICP, which is an iterative, I don't know what it stands for, but basically it's an iterative process in which we try to transform the depth information and try to match it inside within the magnetic resonance volume. And yes, they've been able to obtain really precise measurements. They compared themselves with this system, with another optical tracking system, which is really precise and is really well known in the state of the art. And yes, less than 1 millimeter of error is really, really, really precise. And yes, we are able to fuse that information, get the hyperspectral information, and fuse that and merge that into the magnetic resonance volume. We've done only this in a preclinical stage, but we want to do that inside the operating room. So now I'm going to talk about the machine learning approaches that we follow for biomedical applications. I will talk about ensemble learning. I don't know if you are familiar with ensemble learning or machine learning. However, just to keep things brief, ensemble learning is basically using multiple machine learning algorithms at the same time, so that each of them can predict whatever you want to predict, in this case, brain images, and each of one would give different results. Then you merge the classification for all of the algorithms, and you get a more powerful classification rather than using only one algorithm. So I'm going to show you the results that we are able to obtain, and just to illustrate how powerful ensemble learning can be for biomedical applications. Just before that, I just want to show you a little bit of context of how we are able to optimize our machine learning models. So here, what you can see here is a ground truth. This ground truth is labeled by neurosurgeons, and with different pixels, with different colors, you are able to see different tissues. So in green, this would illustrate healthy tissue. In red, it would indicate pathological tissue. In blue, it would illustrate either blood vessels or arterial vessels. And in pink, it would illustrate meninges. So what we have found is that if we don't optimize the hyperparameters of our models, we are not able to determine where the tumor is. As you can see here, inside this black circle, there should be red pixels, but the non-optimized algorithm, in this case an SVM, we are not able to detect tumor. So one of the things that is really important when using machine learning algorithms for, in this case, biomedical applications, is to perform a hyperparameter optimization, which basically is optimizing some variables that you can configure before training the algorithm. That's what we call hyperparameters. So now, if we optimize this algorithm, this SVM, we are able to detect the tumor. So this, again, is one of the techniques that we use before training our algorithms, because that ensures that at least we can optimize our algorithms in such a way that we can potentially classify better future images not seen by the algorithm in the past or during the training process. Now I'm going to introduce you to one algorithm that we have developed in our research center. It's called Libra. It stands for Low Spectral Resolution Brain Tumor Classifier for Medical Hyperspectral Imaging. We've been working for almost 2 years on this software and also in the paper describing this algorithm. And unfortunately, we don't even have a preprint yet because we want to make sure that all that we are showing or publishing is really well written. And however, I want to introduce you how this works in a really easy way. So let's say we have healthy tumor and blood vessels, okay? We are going to split all of the data that we have into groups of data. So we have our training set. And as you can see here in the training set, we have different healthy pixel groups. So as you can see here, there's one here, there's another one here, another one here, and the same for the other type of tissues. So why do we create these pixel groups? Because if we merge these pixels into small groups, we can then conduct a filtering procedure. And this filtering is basically used to determine which pixel groups are not going to be used for training, maybe because they have noise, maybe because they don't add anything to the machine learning algorithm itself, and also to indicate which pixel groups should go to the different algorithms. So again, ensemble learning means different machine learning algorithms. In Libra, we have 3 SVMs. So as you can see here, in this filtering procedure, we have different numbers. If there's a 0, it means that pixel group is going to be discarded, maybe because it has some noise. If it has a 1 or 2 or 3, it will go to SVM 1, 2, or 3. So we have this huge training set. We create some small subsets of data, and each of these subsets train a different SVM. All of them what we call Libra, okay? So now we train these 3 SVMs, and we perform a classification on a sample or a data set that has not been used during training. In this case, we call it test set, okay? So let's say that this test set is another patient, another human brain. We pass that information to all of these algorithms, all the 3 SVMs. We get a classification, as you can see here, and then we perform a majority voting, which basically is merging the information from the 3 SVMs and get one single result that we can interpret. Once we have that, we have a score. And this score is the one that we use to determine how well this filtering is, how powerful it is to train our algorithm. Now, we indicate we want to maximize such metric so that we are able to find the best filter mask, as we call it here. And we conduct this filtering, Bayesian training, classification, maximize metric, iteratively. So we perform an optimization procedure in where we find the best subsets of data to train each of the 3 SVMs. I know it might be a bit confusing, but this is a really powerful way in which we are able to identify different aspects and also classify different brain tissues. So now I want to illustrate you the results. So we are going to compare an SVM, which is a really well-known classical algorithm from the state of the art. We will compare Libra also with what we call the helicoid classifier, which is basically another classifier that uses one SVM and do some other things to classify data. And these are the results. Yes, briefly, this is the ground truth, as I showed you before. This is the image of the brain of the same patient. And as you can see here, if we try to look at where the tumor should be classified, not even the SVM or the helicoid classifiers are able to locate where the tumor is. However, if we use Libra and we train different SVMs at the same time by performing this filtering that I've described earlier, we are able to locate where the tumor is. So the first row basically indicates with which kind of tissue is, so green would indicate healthy tissue, red would indicate tumor tissue, and in blue it would indicate blood vessels. And in the row below, it's basically these classification maps, but with a k-means algorithm, which basically is used to smooth the classification. Okay, so for instance, if you see here, we can see some granularity, we can see some pixels, but if we apply the k-means algorithms, we kind of apply a filtering technique to smooth all the results. So now, I guess people here is probably familiar with SFDI, and I want to illustrate how Libra, this assembler algorithm, is performing with SFDI data. So previous works have used pig burns, and they've mixed all the pixels together, and the results are pretty good. However, when training machine learning models for biomedical purposes, you should avoid biasing the algorithm. This means that if you are going to classify one subject, you should avoid using that subject in the training process of the algorithm. Okay, so for this experiment, this preliminary experiment that I've been able to do for these past 2 months, I used 2 pigs. One of them, both of them had some burns induced in their skin. And the procedure that I follow was basically, I wanted to train one model with pig, in this case, pig number 2. Pig number 2 has different classes, so skin pixels, pixels that have not been recovered after 28 days, and some pixels that have been healed. Then we train this model with only this pig, and we classify another pig, in this case, pig 3, okay? And then we get some metrics, and we classify the burns so that we can subjectively see how well the model is performing with this kind of maps. And then we also did the same, but the other way around. So we trained the model on pig 3 and then classify pig 2. So we have 2 results. And these are the results. So I'm going to present here the classification maps, 24-hour post-burn. So here is the images, the SFDI images that we are able to use to reconstruct these pseudo-RGB images. These are from fully healed, partially healed, and unhealed burns from these pigs. And this is the end result. This is what these burns would look like in 28 days. And what we want to do is to classify this image on day 1 to potentially see how close the classification is compared to day 28, okay? So the main algorithm, the classical one, is SVM. The results following this methodology with SVM are the following ones. We have 2 systems. So we have Oximager and Clarify. Both are 2 SFDI systems. Oximager allows us to acquire more spatial and spectral information, and Clarify is way more faster than Oximager. So we are using both systems to illustrate how well machine learning models behave under these 2 systems. So what we can see is that the classification maps, although they kind of relate to what they will look the burns in 28th day, the classification maps are quite noisy. However, if we use other ensemble algorithms, a really simple one is random forest, which is basically a forest with lots of decision trees. So many different algorithms, so like 100 decision trees. And they are trained applying some randomness to the data so that each tree is trained on different subsets of data, as I've described with Libra, okay? And if we use this algorithm and we do the same procedure and compare the results with SVM, we start to see that the classification maps are way noisier than SVM, right? And we are able also to illustrate that the algorithm is able to identify where the unhealed skin is and where the healed skin is from just day 1 compared to day 28. And now here are the results with Libra, which is the algorithm that I've described that we've developed in our research center. And the results are even less noisier because we are applying here a filtering procedure, as I described earlier. And the thing that I want you to show in this slide is that by using ensemble algorithms, we are able to get more powerful results rather than using one single algorithm, such as SVM. And that's a really, really powerful thing to use for biomedical applications. So now I've only shown you the subjective results, the classification results, the classification map, sorry. I want to now show you the objective results. So in green, we have random forest algorithm. In blue, we have SVM. And in red, we have Libra. So we have here the results for the F1 score, which is it's a harmonic mean between precision and sensitivity. And we really like to use this metric because it really correlates the objective results with the classification maps, the subjective results. If you use other metrics such as accuracy, they might be over-optimistic when you compare those results with the classification maps. And also F1 score is a really well-known metric used in the state of the art, especially in biomedical, for biomedical applications. So as we can see here, what we are able to see is in each of the columns, we are showing the mean result that we have been able to obtain. So we train model on peak 2 and then classify peak 3. That's one result. Then we train with peak 3 and classify peak 2. That's another result. We compute the mean of those results, and that's what we are showing with the bars. And then you have the confidence interval, which is a confidence interval of 95%, which allows us to understand how the results are distributed, so how well the results are for both cases. So the main thing is that Libra and random forest perform really well when identifying which pixels are going to be healed. And yes, again, using ensemble algorithms is a really powerful thing to use for biomedical applications. So now I'm going to talk about the prototype that I've developed and the main reason why I'm here. I've basically developed an SFDI and a hyperspectral prototype that we are able to measure in the near infrared spectrum. And this is because when we try to classify brain tumors, we basically use homogeneous illumination. And we found, Dr. Dorkin group found out that if you use a structured illumination and apply that information using SFDI to machine learning, you are able to get way better results compared to algorithms trained on homogeneous data, on homogeneous illumination, sorry. So that's the motivation why we want to use SFDI for brain tumor detection. So basically we use a light source with some fiber optics. We apply homogeneous illumination in the brain. We obtain that information with a hyperspectral camera and we train our machine learning algorithms. Now, what happens if we just simply change the illumination? So by that, what we are going to use is a projector to project different patterns of structural patterns, then illuminate the brain surface, gather that information with the hyperspectral camera and train machine learning algorithms. So that way we are able to compare how well the machine learning algorithms delineate tumor when illuminating in different ways, either homogeneous or with a structured pattern illumination. So I'm going to introduce you to my prototype. It's a really compact prototype. I call it HASDI, which stands for Hyperspectral Acquisition for Spatial Frequency Domain Imaging. So I've developed also an interface that allows us to acquire and process data. As you can see here, this is a demo. But basically, we have here the hyperspectral camera. Sorry, this is the snapshot camera that we are using. We have some linear polarizers that allows us to cross-polarize the light and the information that we are gathering for our camera. Then I have here a time-of-flight sensor that allows us to measure how far we are from the prototype to the sample. That way we can configure the projection of the patterns, and that's a really flexible thing to do, to project the width of the patterns so that we are able to project the right spatial frequency. And back here is the DLP itself. So yeah, whatever. We are able to acquire the data, we are able to demodulate everything, and we are actually able to use this in the operating room for pig burns, and it's been used for the past month. And yes, we are getting results, and let's see how powerful this prototype is, because it's quite small, and it might be interesting if we find really interesting results to apply this prototype instead of for pig burns for brain tumors. So I haven't tested my system yet, apart from with the pigs. But before coming to here, I wanted to test the differences that I get in the diffuse reflectance with my camera just by changing the illumination. So for that, what I attempted was to differentiate 4 different substances. So all of these substances, sugar, the salt, baking soda, and polymer, for the eye, they are white, right? It's easy. I wouldn't say it's easy to identify which kind of substance is each one, especially for sugar and salt. And if maybe for us it might be a little bit easy, but for a machine learning model might not be easy. And I wanted to evaluate how well, what's the difference between using homogeneous against structured light. So that's what I've basically done. So we have here to the left an homogeneous illumination of these samples, and to the right a sine wave projected to these samples. So when we obtain this hyperspectral information using homogeneous light, since all of these substances are white, the diffuse reflectance that we obtain across the spectrum apparently look the same. So it's quite hard for the algorithm to determine which substance it's one. However, if we use a simple sine wave, we are starting to see differences in the reflectance. And this might be really helpful, because if we are able, this means that if we are able to apply this in the brain surfaces, maybe we are able to differentiate the healthy tissue from the pathological tissue just by looking at the reflectance. So, of course, we need to use machine learning algorithms, right? So I made a really basic experiment. So I took these, all of these substances, put them all together in a plate, and I used some regions of interest for each of these substances, and get those pixels to train a random forest algorithm, which is a really simple algorithm, again, based in ensemble learning, sorry. And this is the test image. So again, you can see here the different substances that I've used in a more controlled way. So we have baking soda, sugar, salt, 3 different kind of flours, cocoa, Nesquik, and also coffee, okay? So this is the result. So this is the reference. These are the labeled pixels, right, from the test image. And if we train the random forest algorithm when the samples have been illuminated homogeneously, the algorithm don't know what he's actually looking at. He's confused. He thinks everything is flour, or maybe it's cocoa. So he is definitely confused. However, if we use structured illumination, the algorithm is starting to see some differences. Even though they are not really, really precise, we are starting to see something, right? Just by changing the illumination. And again, this is just by using one image to train the algorithm. What happens if we use many, many more images and we use this process of illuminating with a structured illumination? Maybe we can use this information and train more robust models, potentially for brain tumor delineation. So that's the end of the presentation. Before ending this presentation, I would like to thank Dr. Dorkin and Dr. Choi for inviting me here to come to Be Alive to do my PhD stay. Also, I would like to thank Dr. Kennedy and Dr. Campbell, because they are actually helping me throughout my stay to learn more about SFDI and getting more into the details of how it works. So really, thanks about that. And also, I want to thank my university, Universidad Politécnica de Madrid, or Technical University of Madrid, because thanks to them, they've been funding my stay, thanks to these grants for research and teaching stays in the US program that they have. So I would really like to thank them for that. So yes, that's the end of the presentation. Thank you so much for your attention. And if you have any question, I will be more than happy to answer them.
Thank you, Alberto. We're open to questions. got 5 or 6 minutes.
Yeah, thank you for clarification. I just wonder what could be the principal mechanism that as the research shows different with the SFAI pattern lines and the homogeneous.
Like the basics of why it works. You got me that. Okay, I'm trying my best to answer that question. Okay, I'm really new to SFDI, but the main thing is that if you project stripes of illumination, you are basically kind of applying some spatial filterings in the sense that you are not emitting photons in the shadows. That way, photons can go much deeper into the tissue, and you can interrogate more the tissue itself, more or less. Maybe I'm wrong.
I think, the problem with trying to do multispectral imaging to obtain some sort of optical properties is there's always the issue of determining why you don't detect light. Has it been scattered away from the camera, or has it actually been absorbed? And so you have these two parameters of absorption and scattering, and you can't really determine those with planar homogeneous illumination. Whereas structured illumination, the change in the detected pattern that you see will be dependent on the scattering and the absorption in slightly different ways, and so you can tease out those.
Thank you.
You're welcome.
So if you were to build a clinical system to go in the hospital, combine all this stuff, how would it work?
How would it work? We have some ideas. One of them is to use VR glasses or goggles so that they can be attached to the head of the neurosurgeons, and in the glass they are able to see this interactive immersive thing. That should be connected to obviously a computer, a processing station that processes all the information. So the prototype that we have right now is basically for acquiring data. We are able also to show the results, but the end goal would be to show in a screen or in goggles or something the results so that neurosurgeons can navigate through the scene and have that information fused.
Well, first of all, I wanted to just say congratulations on just the breadth of stuff that you and your colleagues that might be able to accomplish. It's really impressive, especially the database. That's awesome.
Thanks.
So I have two kind of related questions. So the first one, I guess, would be: have you looked? So you get a lot of data, right? I don't remember how many wavelengths you get. But you get
400 wavelengths for the line scan and 25 for the snapshot one.
Have you gone through the data to figure out what you don't need?
Yes.
Okay. Like, what's the simplest system you could build that would actually be able to still do what you can do?
That's a really tough question to ask. But the thing is that what we try to do is apply genetic algorithms to proceed some optimization processes to remove those bands that are not necessary. I've been doing this for the past 2 weeks, and specifically what we are able to see is that we need information in the visible and in the near infrared for the brain tumors, not only in the near infrared. And the thing is that it's really hard to determine which are the most relevant spectral bands. And one way to do that is, again, by using genetic algorithms to, in an iterative process, select which ones are the most efficient one for machine learning algorithms. And I know there are some other ways to do that, but that's the way we wanted to go. Also, we wanted to look or get or extract those bands that are closest to the chromophore peaks, the absorption chromophore peaks. However, if we only take those spectral bands, the algorithms are not able to properly identify what's tumor and what is not tumor. So we have 400 bands. It's a lot of data. And unfortunately, with the more data, the more time it takes to train the algorithms. And it's still a work in progress to determine which ones are the most relevant for this case. And also it changes from case to case. So if you are in another biomedical solution or you want to do or apply another biomedical algorithm to another biomedical task, you need to conduct the same process. And it really depends on which camera you are using, how the camera has been acquired, and it really depends. So it's not really easy to say, no, these exact wavelengths are crucial for machine learning algorithms for this specific task. So yeah, it's quite hard to reply to that. Do you have another question? Sorry.
Yeah. So the Libra algorithm or the approach.
Yes.
So the one thing I don't think I quite follow was the filtering part.
Okay.
So I guess is that manually determined or is it a automated process or how that's going to be a pretty critical part of the algorithm.
Yes, it's a really the most important thing of the algorithm, I would say. So so the thing is that we use a procedure that. So we basically use genetic algorithms to do this, right? So we get a genotype indicating which genome should go to each of these algorithms. And this genetic algorithm is doing this process multiple times. So we have several generations, and each generation has a population size. So the better the individual, those individuals would get to the next populations. And we follow that approach for filtering the data. And we have to run multiple iterations to find the most appropriate genotype, in this case, as a filtering mask. So we don't do that manually. That would be really, really hard to determine. So that's why we use that. Unfortunately, we really want to show this because it's really powerful. And once the paper is available, I will let you know because it's really interesting.
One last question.
Yes, please.
Okay. So for the tumor, how the physician identify it? You basically visually identify the tumor and then use this to train the model?
You mean how the neurosurgeon classifies the...
Right.
So we get the data and then we go after the after the surgery, and we ask them, hey, this is the image that you've been seeing. Where do you think the tumor is? And they use their knowledge, the past knowledge, and also the information they get from the magnetic resonance. So unfortunately, maybe the ground truths are not really precise, because they might introduce errors from the neurosurgeons, because I don't know, they even have troubles and issues identifying where the tumor is. So they try to be as precise as possible, and that's the reason why don't we have a really huge ground truth. We have sparse pixels across the ground truth, and that's one of the downsides of this. But it's obvious, it's really hard for them to know where is the tumor, because otherwise they wouldn't need this, right? So, yeah.
My second question, does this system only apply to superficial tumor?
Yes.
Because you get a 2D marker, right?
That's it. That's it.
Thank you. Okay, well, it's 2 minutes past 1. Alberto will be around here afterwards.
Of course.
He's also around BLI if you think of questions. You should thank him once more.
You're welcome.
Yeah, I think I need